Mike from Göttingen, and today I'm going to present to you the effects of epigenetics on learning and memory during aging. And this is kind of uncharted territory in science yet. It's just a new field. And I'll start by something actually very familiar. So, if you look at this young guy, and you give him enough time, well, you cannot really accurately predict what will happen, but there are several options, okay? With time, you will definitely see changes in the brain, which will affect his learning and cognitive functions. We hope that though it doesn't happen, if SENS works. But since it is a general concept that it is inevitable and it happens to every one of us, we are not looking for a specific mutation in a protein, rather for general processes that happens in each and every one of us. So the first candidate will be changes in gene expression. And luckily there was a paper in Nature that's showing that in the age of 40 or 45 years old, if you look at the time of increased aging, so you see at this age, the profile of gene expression is changing. And this is a very interesting age because, well, I don't know, but it's rumored that in this age, you will have a bit of decline in your associative memory and memory function. So what can affect this process? So we are looking at epigenetic mechanisms. So these are mechanisms that affect gene expression without changing the DNA sequence itself. And we turned our eyes to histones, which interact with the DNA and changes the availability of DNA to transcription and therefore expression. And if you look closely at histones, this interaction is very dynamic, and the histones themselves have lots of different modifications that alter the gene expression. And to be more precise, we will look at histone acetylation. And the story is very simple. If you take histone and you acetylate it, you open the chromatin structure, the DNA structure, and this enables the transcription machinery to work. And this is actually a very cool concept because acetylation, if someone wants to ask me later, is also linked to metabolism. So you have a clear link between metabolism and DNA. So that's obviously something that can go wrong in aging, right? So one last thing of my introduction is epigenetic mechanisms are involved in many, many diseases, including Alzheimer's. So actually the question is very simple, what happens in aging? Does it also play a big role? So we started with something very simple. This is a test called the field condition test, and the concept is very, very simple. You take a mouse, you take it very gently in a new cage, and you let it run for 3 minutes. It explores, and after 3 minutes you give it 2 seconds of electrical shock. It's very, very mild, so don't worry. And after 24 hours, if you test the mouse no longer explodes, rather it freezes or shivering. So it learns to associate the shock with the cage. And this is the consolidation of memories appearing these 24 hours. So, as you can probably bet, if you look at young mice, so you can quantify this freezing as time, okay? So 50% of the time the young mice freeze, but when they reach 16 months old, which is not really old mice, but the beginning of aging, you'll see that they freeze much less. So, what can explain it? Well, not morphology, because the morphological structure of the hippocampus, which is very important for short memory formation, there is no difference between the young and the old, and we tested various markers. This is just a few examples. And actually, surprisingly maybe, the levels of histone acetylation between the young and the old mice, there is no difference. It's rather the same profile. So luckily for us, there was already a paper showing if you take these mice and you isolate the tissue of the hippocampus after 1 hour, then you see an increase in young mice. So histone 3 and histone 4, the total acetylation of these histones, after 1 hour field condition, it goes up. So we decided to be a bit more sophisticated, and we did 3 things. First of all, we did a timeline. So we took control mice and 10 minutes, 30 minutes, and 60 minutes. And then we broke it down. So we didn't take the total global histone acetylation, but we looked at specific residues that can be modified with acetylation. So we chose 6 of them, and we also now asked ourselves what happened in aging. And the result is quite striking. So if you just focus on the green bars, you will see that after 1 hour, almost all the histone acetylation go up in the young. And also almost all of them go up in the old, except for one guy, H4K12. So histone 4 in the 12th residue is behaving differently. It goes up in 3 months. It doesn't go up at all in the old guys. So just remember this guy. We're going to talk about it shortly. Why is it so special? Before that, we needed to link it to gene expression. So we did a gene array. And this time we focused only on 1 hour of field condition versus the control guys, and young and old. And we saw something very interesting. If you compare naive mice between 3 months old and 16 months old, there is no difference in gene expression. They are very similar, so the starting point is almost identical. However, if you challenge a mouse, after 1 hour, you see in the 3 months old guys, you have change of the gene profile, so 2,000 genes changing. And the striking thing, if you look at old mice now, there is no response. So there is no elevation of gene expression. And it was such a surprise, we needed to confirm it with real-time PCR with 3 different groups, and we got confirmation. And when we analyze it, it fits the data really nice, because the pathways that involved in this gene expression change connected to exon guidance, long-term potentiation and regulation of actin cytoskeleton. So we have a whole machinery to rebuild or build something or to associate something new.